First of all, thank you very much. I think it's a pleasure to be here, and I can see a lot of great science being discussed in this conference. Since there are many Professor Acharyas in my family, I didn't need to be one, but I will still present because that gene is probably still there in me. So, how many of you have had people who lived 100 and above in your families? A few. So what we did was, and I think all of you know that I'll talk about my grandmom, right, grandmother-in-law. So she lived up to 105, 6. We don't know exactly how old she was. She was, when we looked at her genes, clearly there were some good genes she had. She had incredible habits. I mean, I never saw her eat more than 2 meals a day. And she was constantly engaged in many different activities. She died because she wanted to die after she met all her great-grandkids and so on. But there was also a third component. I think she had a fantastic doctor, right? So she lived in a place where she had all the right things that sort of went, the genes, the habits, as well as the doctor, right? But there was my grandfather. He lived in Bikaner, and while many people over there are, have diabetes and hypertension pretty early in life, he had no doctor. He was also 102, 103 when he died. He had bad habits, relatively. And so I think there was a difference between both of them, right? So there is something that you find that there are people who can live long without the help of a doctor or without the help of having good habits. But in majority of the cases, I would say she would be like a right kind of a person that if you are able to get to that, that would be good, right? So it's been 12 years since we started MapmyGenome. And we've published a lot of papers in Nature, PLOS, and various other things, right? It's been 25 years since I got into genomics. Ossimum was my first company. So what do we do? I think just as an introduction, I think we were the first to get started on the consumer. People ask me, why consumer? There are better ways. Why don't you go to doctors? I said, doctors need to first understand the value of what we are doing. So that's what we started. And we also do a clinical lab, which is a CAP-accredited lab. Now, I'm not going to discuss this because I think in this conference you've heard a lot about the 12 hallmarks of aging, right? So there are many things that are there. But we said, what role does DNA play in all of this, right? We clearly are seeing that there is a role in long life for DNA. Since majority of what we do is genome-wide association studies and also looking at polygenic risk scores, we said, let us find out if there is something in our genes to be able to do that. So this was the paper we recently published. It was in Nature, NPG Aging. For those of you who haven't read it, you are welcome to read it, but I'm going to explain to you in simple terms what we actually did for this study. So we have the world's second largest cohort of over 60, right? But only 0.4% of them actually reach 85 and above, which is a huge, gap because we don't have enough medical facilities, or maybe there are people who are not, like I mentioned, there's habits, there's doctors, and there is the genes. So I think in India, genetics becomes has a lot more relevance. It's the same thing when we were studying. We are also doing another research study, which is more on looking at entrepreneurship and gender, right? Because if your ecosystem, which is all the entire thing, is not there, then your genes are likely to have a bigger effect than not. So I said, is there a secret hidden in their genes, right, in their DNA? Now, how did this idea come about? And you're wondering why I have blue tea and gupshup over here. Because I think most ideas and good research ideas come from chit-chat, right? It comes because you are talking to people, you are able to generate ideas, and that's how good ideas come. So we are having some conversation about tea and expensive tea and antioxidants and living long. And then Professor Gupta told me that, here is a packet, you can take this, and we were having a cup of blue tea. And in that time, we found out that he had published a book on centenarians. So I said, oh, that sounds great. Can we find out the genetic, of all of them? So he said, I don't know how many of them are alive, right? Because that was a few years old. But I said, can you get the percentage of them for me? And he said, yeah, of course, we can get that. So I think that's how the discussion started. And maybe for all the women in this room, I think you will find that one of the things he told me, and I think it always gets a lot of people excited, is that women obviously live longer, but he said something else. He said stubborn women live longer. So I think, which was a great thing for that we found, right? Yes, absolutely. I'll add a third component to it after I show you what we found in the research study, right? So who did we include? Now, initially our plan was 100, then we went to 90, and then finally went to 85 because we couldn't get enough samples at that time. The oldest was a self-reported yoga guru at 126. We also had a family of siblings, 4 siblings who were over 90. And the controls were aged between 18 and 49, right? Who did we exclude? We excluded those that the genotype call rate was less than 98, if there was a mismatch between the gender, so the quality control, or outlier samples that had 3 standard deviations beyond what was acceptable from the top 5 principal components. And then we also looked at the kinship, so how closely were they related, because we didn't want to take too many, any more than one from the same family. So like I said, we had a family of 4. We took one of them. But that was a very interesting set of family to be able to understand what happened in that particular family, right? So we looked at the variants. We removed some of the ones that had low genotype rate. We also removed the ones that had linkage disequilibrium with R square greater than 0.9. And then we confirmed that the ethnicity was actually Indian, so that we were not actually getting samples that were non-Indian as well. Right? We then did association analysis, and I can go into details, but basically what we found was that we wanted to make sure that statistically we were getting a good quality of data set that we had actually collected. So we basically looked at that we wanted to make sure that the variants that we had at least 100,000 permutations to cross this test statistics that we had. So you're looking at a p-value of 5 into 10 to the power of -4 in the logistic regression that we were doing. So this was the p-value that we then looked at as a cutoff, right? So 5 into 10 to the power of -4 after we did all the cleanup and everything else. So we said, can we now identify some interesting genetic variants? So this was what we got. And this is the plot. You can see these are the chromosomes. And that's your p-value log 10 of the p-values. And these are the ones that you can see are the ones that sort of are outside and are more relevant, right? So we said, okay, these look interesting, some not as interesting as the others. So let's dive deeper into these particular variants. So how do they actually relate to an actual pathway, right? So you find a RSID. What does this actually mean, right? Is there some actual correlation between our bodily functions to what we are seeing? Because then it becomes more translatable in terms of what you can actually do. So we found that there were significant SNPs. We mapped them to the KEV pathways. And once we mapped them, we were able to understand some of that information. So this is just the number of samples that we had. After cleaning, we obviously removed some of them. But you can see that the total sample size was about 1,360 samples, 141 cases, and 1,219 controls. So how are these implicated in health? So we found there's some positive correlation and some negative. And for many of you in this room, you will find that they're very well, you can actually see that people who have a lower heart rate, for instance, a lower heart rate marker that we found was positively correlated. Lower risk of bone fractures was positively correlated. Short body height was positively correlated. So I was saying that women, stubborn, and short. Those are the 3 things that you want to look at. So lower heart rate genetic variant, right? So when you have a variant, what does it mean? Now, we saw that there is a variant that has lower resting heart rate. We know there is a physiological impact to that with decreased heart rate. There is a prevalence of about 15 to 20% in the population. And that is linked to lower risk of hypertension, that's linked to lower heart failure, and so on. So I think, for anybody in this room, you know that is there, but now you are able to actually correlate it well with longevity. So we said this can show that if you actually can find means to lower it, that can be beneficial for you. Now, many people get worried, what if I don't have the variant? There are means to counter that effect, right? So you can do meditation, you can look at medication, or you can look at modifications, right? So these are the different things you can do to actually be able to keep that low, for instance. Then you have negative correlations. You have osteoporosis, you have anxiety and neuroticism. And then you have 3 SNPs that were there, but we didn't find that they were as relevant, right? But anybody who's doing the next study over here, you can look at some of these to see if there is a real correlation. So we found some RSIDs that from autism, pancreatic cancer, and celiac disease that were also negatively correlated. But the statistical significance of that was a little bit lower than the rest of them. So obviously, many people have been talking about this FOXO, this particular mutation that is there, because it has been pretty much there in every longevity study that you see. We were very, very excited about finding any relevance that we could find, but we found there is a correlation, but in our study, it was nominal significance, not like you have that and you're all set. What about the APOE gene, right? There was one where we said this is associated with cognitive decline and aging. So this has had very low frequency in the LLIs are long-living Indians, right? So my question was that, when you look at some of the things that you're looking at, osteoporosis, many other things, and especially for women or even for men, is hormone therapy a potential solution, right? So this is a question, I don't know the answer. But this is another thing that I thought that, because this is implicated, is that something we need to study further as well? So basically what we found in our longevity study is that genes do have an effect. Habits matter a lot. Your lower risk for cardiovascular and neuro diseases plays a big role, and gender makes a big difference. So this is the team that did the studies. Dr. Sandhya is our primary investigator. She does all the hard work. I do the fun things of finding out what can be interesting, and there all the rest of the team. Professor Gupta was also involved in this. So now, I've talked about, we did this study, there are certain variants that are there. So if you find out, for instance, that you don't have those variants, is that a problem, right? It's not, because if you want to slow your heart rate, you do exercise, you can have a healthy diet, you can add fish to your diet, you can breathe, you can avoid stress, you can limit alcohol intake. Similarly for osteoporosis and for reducing anxiety, right? So there are many ways that people can actually, if they know, make sure that they actually take those necessary lifestyle interventions. So the question really is, how do you check for these variants, right? So if anyone over here wants to check for these variants, how do you check for that? So that's what we can do, right? You can get a genome patri and you can get these variants. So what are the kinds of things we can give you in that? It would be about healthy aging, telomere length, slower heart rate, neuroticism, osteoporosis, and biliary diseases. And those would show whether those are correlated to your longevity or not. But the idea is not to tell you that if you don't have it, don't be scared. But the idea is that if you have it, what should you do about it, right? So how do you collect these samples? I think most of you are probably already aware, but you can actually do it with just a simple saliva sample. But you want to test the effects of lifestyle, then what you do is you take your family history, you take your genetic history, and then you correlate that with either a microbiome and an epigenetics if you're very excited about that, right? So these are things that you can do today. So you have saliva samples, you have stool samples, and you have blood samples, which is what you can do. You can look at many other things that are you predisposed to something or not. And again, as I tell people, you don't do these things to find out that you're going to die of something, but it is more to be able to help you understand if you are at risk, what does that risk mean? What can you do to counter that risk, right? So you get information about just traits, behavior, and risk of diseases so that you can basically do that. Now, when you look at other such risks, whether it's type 2 diabetes or not, this was another paper we had published in Nature. This was about how Indians actually are likely to be at risk for type 2 diabetes or not. So again, I think many of you are already aware, but if you are wanting to understand about your gut health and all, I think none of them can be taken in isolation, right? I know that we have had some discussion in this conference where we said epigenetics doesn't work or microbiome doesn't work or something doesn't work. I think in the end, health is complex nothing is a single on and off switch. So it's complex, and therefore you need to understand things that are not just baseline, which is your genetics, but understanding your family history, changing your diet and lifestyle, and making sure all of that is there. And these are tools to help you get there. But until people actually do it, none of this is useful. So most important, look at your genes, because you need to get the right doctor, and you need to make sure that you have the right kind of habits and actionability that you can do yourself. So you can, this is a gut score that you can get, and you can get epigenetic scores if you're looking at it. You get counseling, which is the most important piece, because unless you do counseling, most people will not understand the value of whatever you're doing. So it's not like looking at a risk score, not about looking at a particular gut microbiome, but it's about putting all these pieces together for one person. So with that, happy to take any questions that you may have.
This is the kind of very quickly.
I think we talked about before the dinner. What about the other genome, the mitochondrial genome?
We do look at the mitochondrial genome, but usually that is in a clinical setting.
Yeah, but the reason I bring it up because Masai Tanaka has been published from 25 years ago, and then Claudio Franceschi published that, and they found in centenarians and supercentenarians there was a correlation between the mitochondrial genome and the variants in the nuclear genome because of the co-evolution of those two genomes. So even if we have, say, FOXO3, or you don't have that FOXO3A, but if we have good mitochondrial variants, you may be okay.
Yeah.
So is it in the pipeline, or you going to do it?
We can do it. So that's why we have these dialogues, right? So, yeah.
Yes, and what is the best?
Yeah.
Can I coordinate this? The gentleman there.
Okay, he first.
And what is the, in your experience, what are the best resting heartbeat? And what is the cost of these tests?
Oh, the tests are right now quite cheap. They are about 5,000 or so, but it's typically about 7,000 to 8,000.
Okay. Are you in Bangalore? It is available. This test?
It's available all across India and across the world as well.
And what is the best heart rate, the resting heartbeat?
I think it depends on the person, but resting heart rate, lower heart rate typically is good. So for instance, mine is 49, but I know people have 38, 40 also, but I have some of good genes, but I also have very lazy genes. So I don't do all the things that I preach, but I think resting heart rate is an important part to calculate some of your, if you go to our website, there's a fun calculator that you can just put in your resting heart rate. You can put in your number of steps and a few other parameters, and you'll get some sort of an estimate. It is not accurate. It's more for just getting you started.
So do you have decade-wise epigenetic signatures of people as to how they're aging?
Very less. I mean, we don't yet have it, but we want to get there. The epigenetics is still relatively new. We work with True Diagnostics right now, so it's not something we do ourselves. Everything else we do on our own. So, and because the test cost is a little bit expensive, not many people do it that often. There are a few clients who have done it 3 or 4 times, but not enough.
Okay, because if you look at people who are long lived, of course there's a visible difference between their overall body structure. For example, a lot of people who are long lived have very low fat mass, as is evident, right? There is sagging and so there has to be some, there has to be some genomic signature which determines how they're going to be over the next, say, 5 years.
Correct.
So it would be interesting to have a longitudinal study of that. So
Any longitudinal study is great, right? I mean, I think that's the important part, that if we can get even your, ideally, I'm hoping that in the next year or two, the pricing of sequencing is going to go down even more. And then I think you will start seeing that this will become like it'll truly spread, because currently I think it is still like your microbiome and things that you do on a repeated basis. Genetics you're doing once, but you are doing the epigenetics or microbiome. I think when the cost becomes more like 3,000-4,000, you're going to see more people doing it much more regularly.
We have the last question.
Since you brought up short stature, did you see any connection with insulin pathway? That also works in canines.
In?
Insulin pathway. So was there any?
Yeah, we were hoping.
If short stature is related.
No, we definitely saw short stature. We did not find that, but I must say many things are they will. I'm just reporting what we saw. And in that, we didn't find those particular genetic variants. But maybe we need to do a bigger study, and maybe we'll find something there.
So with that we'll end. Please let's give a round of applause.
Thank you.